Hello guys, we are gonna talk about chapter 19, which is viruses. So yeah, let's get started. So a few things that we need to know: the components of viruses and different things, how they're basically how they reproduce themselves, how can they make more viruses from just one single virus. Okay, so a few things between bacteria and viruses I want to let you know. Oops, sorry. So, bacteria is on the side. It's actually like a living cell. So even though it's not it's not like super complex like our cell, like human cell, but they're actually living cells that can actually function, like I said right here. And then they're usually looking something like this. They usually have a tail, or they have a lot of more tails. To help them to swim from one side to the other side. So in comparison between bacteria and viruses, later on we'll go into bacteria a lot more. Between them, viruses are actually much smaller. So you can see the scale wise, it's 1,000th size of a bacteria. And actually, for bacteria, a lot of time you hear antibiotic. That's what we use to kill bacteria. But for viruses, basically vaccines only way we can prevent viruses from infecting the host. So viruses is something important. It's actually not a living cell, okay, and it actually act like a parasite, but it can't really survive by itself. So that's a few of the things that's very important. And when we talk about antibiotic, we can also talk about antiviral treatment that's specifically designed for viruses, which we will talk about in just a little bit. So viruses are very small, and as you can see, it's actually smaller than the ribosome. So ribosome is already tiny because it's making RNA into protein. So from that, it's even smaller. Viruses are even smaller in sizes, and viruses actually have three that's major, that's common components that we can see. You have nucleic acid, usually is usually is RNA, but sometimes it's DNA, and then you have double strand or single strand nucleic acid, depending on different kind of viruses. And you also have the capsid, which is the green part right here. Green part basically it's a shell that kind of protects the material, the information, the blueprint of these viruses. And some of viruses also have the viral envelope that helps them to kind of fuse into the cell rather than inject its RNA or DNA into the cell. So that's something very different. Only some of the viruses have. Okay, viruses, because I said it has a parasitic property, it actually requires a host, and it's very specific on where it wants to affect you. So a lot of like cold viruses, like flu, flu, the every single day flu viruses, they actually mostly infect upper respiratory tract. So that's why you have like coughing, mucuses, and etc. And again, because viruses is not alive, it actually requires a host to produce itself, and it can produce very efficiently, which we will talk about next. So these are a few of the different kind of viruses. As you can see, that you have something that's more tubular, and you have something that's more like geometric shape, and then something that's more like circle. And this one, like the previous one, like we said, it has a viral envelope that allows it to fuse into the cell instead of injection. And then since we're talking about injection, this is a viruses that's what we call bacteriophage, and it can actually inject its DNA, excuse me, DNA information right through this tail, and then inject it right into the cells of the host. So they have different different size, and then as you can see, some of them are much smaller but elongated. Some of them are much larger. It depends on what they want to do. And generally, whenever you have a membrane outside, it's going to be slightly larger than the other one. So there are some of the animal viruses right here. You have the double-strand DNA, single-strand DNA, double-strand RNA, and these are just a chart right here. I'm pretty sure. Sorry, I'm pretty sure you guys also have this one. Sorry, I accidentally jumped out the PowerPoint slide. Okay, you also have a single-strand RNA right here, and then you have single-strand RNA that's a template for RNA synthesis. And then basically you have, right here I'm trying to show you, it's different types of viruses that has different kind of information within. Okay, so let's simplify the viral replication cycle. Like I said, viruses actually accumulate by themselves within the host body, but it cannot do it without the host. It needs a host to survive and then to move on to different things. Oops, sorry. So right now we have basically a virus is coming in, and however way that it use, it basically going to enter the host right here, which is this orange area. Okay, and some of them are going to actually lose its capsule, basically, and then have the DNA or the RNA. In this case, it's DNA right here. And then basically, this DNA is going to hijack a lot of its internal component from a cell, and then from that, the DNA or the RNA, in this case, DNA will become, well, basically, it will replicate because it will be hijacked. It will use the ribosome and use the other part to basically replicate the DNA information within the host. And furthermore, this DNA will also be transcribed and translated into protein. And from protein, you can make more capsule protein, which would allow them to have the outside shell. And then when those are available and ready, you will have newly grown viruses, particles, and then eventually when there's large amount, usually when there's huge amount of it, they will all release all together, and then basically a few of them that will actually cause the whole cell to burst, and then basically it release, like, just imagine like you have a whole bunch of balloon from, I don't know, from a fair, and then you have it all piled in the classroom, and then just it's all in here, and then when the teacher comes, open the door, every single balloon just flew out. Basically, it's like that. So that's a way for them to kind of replicate within the host cell. Okay. Let me pause. So this is the next thing. We have bacteriophage. Basically, this is how they inject their information into a cell. So, like, as you can see, it's almost kind of like a spider. Just imagine a spider, and it lands itself onto a cell, and then it basically inject itself into the cell right away directly. So this is a video. I will show you the link. I don't want to waste time right here. Okay, this is a lytic cycle of a T4 phage. So T4 is this kind of bacteriophage, and then basically this is how they reproduce itself. So as you can see, T4 actually has a few different components. You have the head, you have the tail, and you have basically their legs, the tail fibers. So first you have the attachment, like we show right here in this picture. You have attachment, and then from attachment it basically inject its DNA into the host. Okay, and then it's going to, by injecting it, it's going to hijack a whole bunch of different components within the cell that actually degrades the host DNA. So this, imagine it becomes a zombie that would function just like a normal person, I guess, zombie. But this zombie is not actually gonna have thoughts of its own. So you can kind of control it. So this bacteriophage will actually control everything internally and ask every single protein and every single ribosome to make more bacteriophage components. And as it makes more, as you can see, it becomes, well, it's everywhere. It's not very organized. So with all these components, it will actually assemble itself like this. Okay, and with other protein help as well. And basically from that, like I said before, when there is so many of them, all of them are going to release at the same time. And when they do that, basically you destroy the whole cell altogether. And then all these individual viruses are going to further go to the next host cell and then basically perform more destructive replication process. So this is the lytic cycle. Okay, now we're gonna talk about the difference between lytic cycle and lysogenic cycle. And you can imagine lytic cycle as the bacteria just killing its host right away. And the lysogenic cycle is kind of like a dormant period. So what it does, it actually, well, I'm gonna give you guys a few seconds to pause, and I'm gonna go on. Okay, so these two cycles are often used interchangeably in some of the phages. For example, the temperate phage actually use both methods to replicate itself. So this is the difference between lytic cycle and lysogenic cycle. Lysogenic cycle, as you can see, after the bacteriophage inject its DNA, the DNA actually fuse with the host cell. And then as it produces more daughter cells, it will actually also produce this bacteria DNA as well. So once you have, imagine like after this production, instead of one infected cell, now you have two, and then so on and so forth with exponential growth. So eventually it will become so ideal that this DNA would actually come out by itself. Well, actually not really come out by itself, but it actually will be cut off from the original host and then become activated. And then from activation, you thus produce different components of bacteriophage, and then eventually the bacteria becomes assembled, and then they're released all at the same time, and then cell lysis, and basically the cell dies. So this is a kind of combination of lytic cycle and lysogenic cycle. You have the bacteria going in, you have the cell reproducing by itself without knowing that it's been infected, and then all at the same time, all these viruses actually all burst out. And later on, when we talk about cell communication, we will talk about more how do they know when to burst out, basically when to activate, or when to all these things happening. So these are this is an animal virus that have a membrane outside envelope, and how do they do things. So very different from earlier. Earlier only has two layers. You have the information nucleic acid, and you have the capsule, and then now you actually have an envelope. Okay, like I said, envelope allows it to fuse into the cell, so you're not actually destroying the cell, like the lytic cycle that we show right here. This is a destruction. Like it just completely destroyed the cell. This one, you actually fuse with the cell, and you have every single information now inside alongside with your capsule. And then with your capsule, actually you can release the RNA. That RNA will actually go ahead and make more mRNA. And from that mRNA, well, with ER and so on and so forth, would produce the outside, the envelope components, and then let it release into the surface of this host cell. However, if you only have the outside component, it's not going to work. Nothing's going to happen. So you also have the replication of, making its capsule protein. And furthermore, it also allows itself to copy its RNA. So these two will fuse together to form a new inner part with the capsule and the RNA information. And when these are, when they're in great number, or when they're gathered together complete, now we can actually move to the surface. The capsule will move to the surface and then basically butt out into new viruses, and this new virus can go ahead and infect the other one. And obviously, you can see this is the example of one single virus is producing one extra viruses, but in reality, it will produce much more viruses just from this process. This process just repeats itself. And this is very difficult because it can actually land without actually injecting into the host cell. And as long as you can recognize something in the host cell, it will be able to attack. So this is actually something very difficult for our immune system to detect. Sorry.